We are jumping from Turner and Perkins, well, the Turner and Perkins article from 1979, where they demonstrate the existence of Spore Killer 1, Spore Killer 2, and Spore Killer 3. And we are going to skip 23 years and now take a look at one of the articles that I am an author on. So Hammond et al. In 2012, published in PNAS volume 109, starting at page 12,093. So, wow, a lot of articles published in PNAS in 2012. So, just briefly, so remember that Turner and Perkins had found that the genes for spore killing were located spore killing by SK2 and SK3. We are ignoring SK1 for now. So the genes for SK2 and SK3 based spore killing are found on chromosome 3, and they also identified a recombination block or a suppressed region of recombination. Some researchers call it a block, some people call it a region of suppressed recombination. They found this, I like to say blocked, they found this blocked region on chromosome 3 when a cross was performed between an SK2K strain and an SK2S strain, or an SK3K and an SK3S strain. So in these two crosses, they found a region of suppressed recombination between ACR2 and LU1, so these genes on chromosome 3. And they had another gene out here, HIS7, so between LU1 and HIS7, they didn't see this evidence of suppressed recombination or blocked recombination. And remember, recombination is essentially crossing over. So 23 years go by and researchers are still looking at SK2 and SK3, and they discover a lot of important things. So essentially, the region of suppressed recombination is much longer than the interval demarcated by ACR2 and LU1. So I'm going to put another gene out here, cum for cumulus. So when this gene is mutated, the fungus grows in sort of a spiral cloud-like, cumulus cloud-like pattern. And I want to put another gene, AD4, which is required for making adenine. And another, let's see over here, ACR7, which is another gene that confers resistance to the metabolite acroflavin. And so what they find over this 23 years, by trying to identify the genes that control spore killing by SK2 and SK3, they know they're on chromosome 3, but what they now know, after a lot of research during this time, is that the genes required for spore killing by SK2 and SK3 are found somewhere between the cumulus gene and the LU1 gene over here, or around the cumulus and the LU1 gene, and between those two genes. So this giant interval here, that's where the genes required for spore killing are found by spore killer 2 and spore killer 3. And the length of this region, so they don't have the physical length during this 23 years, they don't have the number of base pairs, but they know that in a normal strain, in a cross between normal non-spore killer strains, that this region is 30 centimorgans long, 30 centimorgans. So that what that means essentially is that they will detect 30% crossing over in this interval normally when you don't have an SK2 parent or SK3 parent as part of the cross. And I'm going to estimate, with actually looking this up, I'm going to say there's probably between 500 and 1,000 genes in this region right here. So, another important gene, another important thing they discovered was, remember those strains. In the previous video, we talked about the SK2R strains. So those are strains that are resistant to spore killing by SK2K, but they are not spore killers themselves. And the SK3R strains. So those are strains that are resistant to SK3-based spore killing, but they are not spore killers themselves. So they identified the approximate location of the genes that allow an SK2R strain to be resistant. And they name that, they just move this R down here and put parentheses. They name those locations RSK2 and RSK3. And what they find is that somewhere in here between cumulus and ACR7 is the genetic information, or the genes, RSK2 and RSK3. So they don't identify the sequence of these, they just know that somewhere in this region towards the left border of, well, I don't want to say that, to the left of ACR7 and to the right of cumulus, and who knows, there could be like 100 or 200 genes in here. That's where one should be able to find the RSK2 and RSK3 genes. Okay, and so that's where Hammond et al. 2012 PNAS, that's where this article starts, working with this information. This article is going to try and learn a little bit about how spore killing works. Okay, so what did Hammond et al. 2012 attempt to do? Well, essentially, they were looking at the SK2, or at least they started with an SK2R strain. So this is a strain that is not a spore killer, but it is resistant to spore killing. And this work was made possible because researchers had already identified the approximate location of a gene that makes the SK2R genotype resistant to SK2-based spore killing. And so we're going to refer to the gene, at least at this point in the video, we're going to refer to it as RSK2. And researchers had already shown that if this is chromosome 3 in an SK2R strain, and let's say this is where the cumulus locus is, this is where acroflavin 7 is, this is where ACR2 is, LU1, HIS7. They had already shown that if a researcher wants to try and identify the exact location of this RSK2 gene, well, they should look between cumulus, to the right of cumulus, and to the left of ACR7. And I don't know how many genes are in there exactly, but it could be, let's say, a couple hundred. Let's say 200 to 300 genes in there. So 200, 300 predicted genes in this interval represented by, or bounded by the cumulus gene and the ACR7 gene. So that's a lot of genes. That's a lot of genes. And researchers hadn't identified this gene precisely, and so this is where me, during my postdoctoral fellowship, this is me, and working with other lab members and my postdoctoral advisor, set out to learn about how SK2 possibly works by focusing in on and trying to identify this gene, its sequence, and things like that. Okay, and so how are we going to do that? And that's what I'm going to cover in this video. Okay, so Hammond et al. 2012 focused on a strain of Neurospora crassa called LA222. Now this strain is an SK2R strain, meaning it is resistant to spore killing, but it is not a spore killer itself. And previously, researchers had shown that if this is chromosome 3, well, importantly, there's no recombination suppression with this strain. So there's no recombination blocked or recombination suppressed interval of chromosome 3. So crossing over works normally when, let's say, an SK2R strain is crossed with an SK2S strain. So recombination and crossing over works as expected. And they had shown that the gene that confers resistance to SK2 based spore killing in, let's say, this strain right here, LA2222, is found between Cumulus and ACR7. We don't know exactly what the gene is, the identity of it, the sequence of it, but we know it's somewhere between Cumulus and ACR7. And so what Hammond et al. Did was they wanted to learn more about spore killing, and they thought the easiest thing to do would be to identify the gene precisely, identify the gene, identify its sequence, the sequence of the gene that confers resistance to spore killing by SK2, the gene that makes this strain, LA2222, have the SK2R genotype. And so that gene at the start of the study, or by previous researchers, was named R, parentheses with SK2 in parentheses. Okay, so Hammond et al. Set out to identify this gene. And we already had an approximate location, somewhere between Cumulus and ACR7, and let's just say there are probably hundreds of genes in this region here. So first thing they had to do was refine the location, so narrow down the location to a smaller region. And they used 3-point crosses, and maybe some of you remember 3-point crosses from your introductory genetics courses. And so they used 3-point crosses, and what the 3-point crossing data told them was that this RSK2 gene would be found or exists in an interval that contains 4 predicted genes. So let me step back a moment. In 2003, the Neurospora crassa genome sequence was published, and that genome sequence was for what we would call an SK2S or an SK3S strain. So it was sensitive to spore killing. The genome, they sequenced the genome of a strain that was sensitive to spore killing by SK2 and by SK3. And that genome sequence predicted that there were 4 genes in the region that RSK2 was mapped to, and those genes, it was not possible to predict what their function was at the time, but they were given numbers, database numbers, NCU 09151, NCU 09152, and you can bet the NC, I think it stands for Neurospora crassa, I don't know what the U means, NCU 09153, and NCU 09154. Okay, so the 3-point crossing data suggested that RSK2, what was previously known as RSK2, might be one of these genes in here. Okay, so in the last segment, I had described how Hammond et al. Had used 3-point crossing to refine the location of RSK2 in strain LA2222 to an interval of chromosome 3 in the strain that contains 4 predicted genes, and I'm abbreviating the database numbers to 9151, 9152, 9153, 9154. And essentially what Hammond et al. Did was they deleted these genes one at a time from the LA2222 strain. And what they were able to show is that with a series of crosses is that only when 9151 is deleted does the strain lose the ability to resist spore killing. So let me diagram some of the results here. So let's take our control strain, LA2222, with the SK2R genotype. And then we'll have LA2222 over here. Now this one has the 9151 deletion. I can still put SK2R here for now, but it's this original genotype with 9151 deleted and replaced, and sometimes we put 2 colons here to indicate that the 9151 gene was deleted and replaced with a hygromycin resistance selectable marker. So this is a gene that confers resistance to the antibiotic hygromycin. Okay, so when this strain right here, the control strain, is crossed to an SK2K strain that also is deleted of SAD2. Okay, now this is a gene in Neurospora crassa that controls MSUD. MSUD stands for meiotic silencing by unpaired DNA. And it's a somewhat complicated phenomenon. And for now, and I might put together a video that can help explain this. For now, we can just we can ignore the complications of MSUD by just studying spore killing with at least one of the parent being deleted for this SAD2 gene. When SAD2 is deleted, MSUD doesn't work that well. And it makes it easier to study spore killing. So that's where that SAD2 delta is. And in most of the crosses, many of the crosses, you're going to see there's a SAD2 delta in the cross, and that just suppresses this process. It makes it easier to study spore killing. Okay, so when LA2222 is crossed to the SK2K strain, what we get is the expected asci with 8 black ascospores. And half of the ascospores have the SK2K genotype, and the other half have the SK2R genotype. Okay, that's what we expected. Okay, that's our control cross. Now, of these 4 genes here, in the LA2222 strain, only when we delete 9151 do we get this result over here. Only when we delete that one do we get the result of 4 black ascospores and 4 dead ascospores. The 4 black are the SK2K genotype, and the 4 dead are presumably the SK2R9151 delta genotype. Okay, so this result indicates, right, that when we delete this 9151, that strain that is normally resistant to spore killing is no longer resistant to spore killing. We don't see the same thing when we delete 9153 or 9154. It actually wasn't possible to delete, at least in that study, 9152, and we show by some other complicated processes that 9152 is not required for resistance to spore killing. So 9151 seems to be the key gene here that provides resistance to SK2 based spore killing. Okay, now that we know 9151, the gene NCU09151 is the RSK2 gene. So this is, you can assume this is the common name of the gene, and this is the database name. We have an interesting problem, right? Interesting problem. And perhaps it's best to see this problem through a diagram. Let's take a standard SK2K strain, and why not? We'll make it the SAD2 delta genotype. So MSUD does not work in crosses with this. Again, MSUD, I'll have to have another video on that at some other point, but just know that we're suppressing it to make all these studies easier. So when we cross SK2K to SK2R, we have 8 surviving ascospores with half the SK2K genotype and half the SK2R genotype. When we cross SK2K with SK2S, well, only those ascospores inheriting the SK2K genotype survive, and the SK2S ascospores inheriting SK2S are killed. But the problem is, right, that SK2R has the NCU09151 gene, so does SK2S. It also has the NCU09115 gene. So what is it? What is it about NCU09151 in this strain that makes it resistant, but not in this strain? What is it about this strain's version of NCU09151 that makes it resistant, as compared to this strain's version, which does not make it resistant? So it is the reason is these are different alleles, right? So alleles are different versions of the same gene. So different alleles have different sequences. And so if we should look at the sequence of, let's say, NCU09151 in the SK2S strain, I'll superscript that, it's going to be different than the allele of NCU09151 in the SK2R strain. And if we do a protein translation on these sequences, we can see that the protein sequences are different. So it seems that the allele of NCU09151 determines whether or not the strain is resistant to SK2K or susceptible to SK2K-based spore killing. Okay, so now that we know NCU09151, the specific allele of NCU09151, can make an SK2 or makes an SK2R strain resistant to SK2-based spore killing, we can ask ourselves, okay, what makes SK2K resistant to spore killing, right? Okay, so let's take a closer look at that with a diagram. So SK2K crossed to SK2S. Now we should know the phenotype we're going to see in each ascus from this cross. We're going to see 4 black ascospores and 4 dead white ascospores. So apparently there's a killing process going on that kills these 4 ascospores down here, and these 4 somehow survive the killing process. And so the hypothesis by Hammond et al., once they discovered that this gene right here, NCU09151, confers resistance to spore killing in these strains up here with this genotype, they thought, well, maybe SK2K uses a version of NCU09151 to resist its own killing process. So maybe it makes a poison and it uses a version of 9151 to resist spore killing. And so it turns out it does. It does. So let's see here. So now that we knew the sequence of what an NCU09151 gene was, we're able to use PCR to look inside or look for the gene, the orthologous gene, or the same gene in SK2K, and delete that. And so what happens SK2K, when you delete its allele of NCU 09151, when you delete that, and cross that strain to SK2S, instead of getting this phenotype over here, what you get is empty asci. So no ascospores. So everything dies. Everything dies. So what that tells us is that, oh yeah, okay, SK2K uses 9151 to resist the effects of its own killing process. And when you delete that, everything dies. Everything dies because there's no resistance in that ascus, right? Because the SK2S has 9151, that is, its allele is not resistant to SK2-based spore killing. And when we delete SK2K's version of 9151, okay, well, there's no resistance in that ascus. Everything dies. Everything dies. Now, what about SK3? Well, it turns out SK3K also uses an allele. Of 9151 to resist its killing process. So when we find the NCU 09151 gene inside SK3, the interval, remember that recombination blocked interval that contains all the genes required for SK3 spore killing, well, one of those genes is 9151. And when we delete that, we get the same thing that we saw up there. Instead of getting 4 black ascospores with the SK3K genotype and 4 ascospores, dead ascospores, with the SK3S genotype, what we see is empty asci. So no ascospores. Okay, so these results indicate that both SK2K and SK3K use alleles of 9151 to resist their own killing mechanisms. So in summary, Hammond et al. Identified 3 classes of NCU 09151 alleles. So 3 classes. So let's say class 1 is going to be found in strains that are susceptible to SK2 and SK3 based spore killing. So remember in the previous segments of videos and previous videos when we were saying a strain had the SK2S genotype or the SK3S genotype, well, those strains have an allele of NCU 09151 that confers no resistance. Class 1 is no resistance to SK2 or SK3. Class 2, to keep it simple, we can say that class 2 is found in SK2K and SK2R strains. So in the SK2K killing strains, in the SK2R resistant to SK2K strains, those strains have the class 2 allele of NCU 09151. So those alleles resist SK2 based spore killing. And the final class is class 3. The class 3 alleles are found in the SK3K genotypes and the SK3R genotypes. Now these resist SK3 based spore killing. And what makes an allele a class 1 or a class 2 or a class 3? It's a sequence. The sequence of the allele and the resulting sequence of the protein that is encoded by the allele is what makes a specific version of 09151 not resistant to SK2 or SK3, resistant to SK2 or resistant to SK3. Okay, so finally, Hammond et al. Proposed a model for how spore killing works, at least with SK2 and SK3. And that model is called the killer neutralization model. Well, the killer neutralization model of spore killing. And essentially, it assumes that spore killing works through a poison and antidote system. And taking SK2 and SK3 as our examples. So SK2 is presumed to make a poison. Let's say that poison has a certain structure. I'm going to make it a circle. And SK3 makes a different poison. I'm going to make it a square. Now, SK2 makes an antidote. Hammond et al. Suggests that that antidote is a version of 9151, a protein encoded by the NCU09151 gene, specifically the SK2 allele, or the allele found in SK2K strains might be an example. And so what that antidote will do is it will bind the poison and neutralize it at specific stages, during specific stages of meiosis. And so let's say this antidote protein looks like that. Now in SK3, the antidote protein is going to be a different version of NCU09151. The SK3K version of that, and it has a structure that looks like this. And it is going to neutralize the SK3 poison. So the SK3 antidote cannot neutralize the SK2 poison, and the SK2 antidote cannot neutralize the SK3 poison. And that would explain why when SK2K is crossed to SK3K, that everything dies. Everything dies. Oh, okay, that's a little too complicated. I need to show you some other aspects of the model before that makes sense. So, okay, so these are the key components of the model. Now, we need to go back to our introductory Neurospora biology video where we talked about how ascus development works and how meiosis progresses at the beginning from the diploid stage to the final mature ascus stage. Now, if you remember, meiosis starts or is initiated by the Crozier hook cells, where we will have haploid nuclei present, one haploid nucleus from each parent of the cross in this penultimate cell of the Crozier hook. Let's say this is a nucleus here from, let's say, an SK2K strain, and let's say this is a nucleus here from the SK2S parent. Now, the SK2K strain makes the poison and the antidote for the poison. And so we can assume for this model, I'm diagramming the antidote here, and it's neutralizing the poison. It's neutralizing the poison. So the cell is alive. This penultimate cell here is alive because the poison is being neutralized. Okay, so meiosis begins. This cell elongates. These nuclei fuse to begin meiosis. And now I will diagram the meiotic cell as elongated to make things easier. And now the nucleus is diploid, containing the genomes of both of these parents. And the chromosomes are pairing up. And the poison is being made because the poison gene, which we don't know the identity of yet, the poison gene is encoded in the SK2K genome. And so there's the poison, and it's being neutralized by the SK2K antidote. And remember, the SK2S makes a version of 9151 that essentially is useless in this cross. It doesn't neutralize the poison. But the poison is neutralized, so this diploid cell is perfectly happy, perfectly happy. Now, the next stage is after the diploid nucleus goes through the first meiotic division. So now we have 2 nuclei. And I'm not going to diagram the poison and the antidote, but the poison and antidote is, as it's here, it's also over here. The poison is being neutralized, so nothing dies. We go through the second meiotic division, so I'm going to call this meiosis 1, this meiosis 2. Oh, I didn't mean to make it meiosis 3. Meiosis 2. So now we have 4 nuclei. Again, the poison is present. It's being neutralized by the antidote. We go through the post-meiotic mitosis, which is something weird that happens in Neurospora to give us 8 nuclei. 1, 2, 3, 4, 5, 6, 7, 8. I'm going to cross that one out. So now we have 8 nuclei. The poison is still present. The antidote is still neutralizing the poison. And now, this final stage, when the walls of each ascospore are being made, there's our nucleus, there's the ascospore. The walls are being made, the ascospores are being cut from the shared cytoplasm. Now remember, half of these ascospores are getting the SK2K genes, and those are the survivors, SK2K, and the others are getting the SK2S genes, and they are the ones that die. So the killer neutralization model holds that only the SK2K ascospores continue to make the antidote, and the antidote is stuck. The antidote protein cannot move out of the ascospores, So they can only neutralize poison that is inside the ascospores. So that these ascospores, the SK2K ascospores, do not succumb to the effects of the poison. However the poison works, whatever it does to kill ascospores, it cannot do that in the SK2K ascospores because they encode the antidote, the version of 9151 that neutralizes the poison. But the antidote, the version that neutralizes the poison, cannot leave the ascospores. But the poison, let's say the poison is really small, it can leak out of the ascospores and it can kill everything else in the ascus. It can kill everything else. And so this poison is not neutralized. And these ascospores down here, they do not encode a resistant version, a version of the antidote that can neutralize the poison. So they all die. They all die. Okay, so that's the killer neutralization model. Now, it also explains why when we do a cross of SK2K 9151 delta, so the 9151 gene inside the SK2K genome, when we delete that and replace it with hygromycin and cross that strain to SK2S, what do we see? Well, everything seems to work just fine. So we have the Crozier hook, we have the nucleus from the SK2K 9151 delta strain, we have the nucleus from the SK2S strain. This becomes the meiotic cell. At this point, Perhaps the poison isn't active at this point, but at this point, the poison becomes active. The very early stage of meiosis, the poison becomes active. We deleted the antidote gene from the SK2K strain, so there's no antidote to neutralize the poison, and so the ascus dies at this stage and no spores are produced. So if you remember, you can go back to earlier parts of the video when we deleted this 9151 delta, when we deleted 9151 from the SK2K strain, did this cross, I said no ascospores were made. And that was a strange phenotype, something that hadn't been seen before. And that explains it. It's possibly explained by the poison not becoming active until this early meiosis. And once it's active, well, there's no antidote around. And so that meiotic cell dies and no ascospores are produced. So the killer neutralization model explains a number of different phenomena that we see with respect to the deletion of 9151 in various strains. Okay, so I think that's it for Hammond et al. 2012. What we're going to see in the next video, so we've shown that SK2K and SK3K use a gene with database number NCU09151 for resistance to spore killing. Now, the common name for this gene used to be RSK2 or RSK3. Before this study, researchers didn't know if these were the same gene. Turns out they are the same gene, just different alleles of the same gene. Now, the common name of this gene is just RSK. So the database name is NCU09151. The common name is RSK for resistance to spore killing. And not all alleles provide resistance to spore killing, only some alleles confer resistance to certain types of spore killing. And now this finding, the finding of RSK, is going to help identify the gene that controls poison production. And so we'll take a look at that in the next video.